Kinetoplastids are unicellular eukaryotic parasites defined by an organelle called the kinetoplastid, the organism's only mitochondria. Examples of kinetoplastids include human-infecting species like African trypanosomes, the causative agent of African sleeping sickness. Crithidia fasciculata is another kinetoplastid containing similar biology and genome, but does not infect humans, therefore making it an excellent model organism. Using Crithidia fasciculata, applicable findings can be made to other kinetoplastids, including apoptosis. Apoptosis, or programmed cell death, for kinetoplastids help regulate the growth of the organism through selfish altruism, the favoring of the fittest parasite for survival and transmission in order to successfully continue the cell cycle in the host vector. Kinetoplastid apoptosis shares hallmark characteristics seen in higher eukaryotic organisms, such as cell shrinkage, chromatin condensation, and DNA fragmentation. But specific genes and pathways that regulate these functions differ between mammalian cells and kinetoplastids. In mammalian apoptosis, the gene apoptosis antagonizing transcription factor, or AATF, has been shown to be a regulator of p53, activating cell death. Once AATF is phosphorylated, it activates p53 transcription, and an increased expression of p53 induces cell cycle arrest. This will prevent the replication of any damaged DNA, thus resulting in the apoptosis of the cell. We have identified an AATF homologue that is present in all kinetoplastids. Studying the function of this gene and its associated pathways will lead to better developments of treatments for pathogenic kinetoplastids. CRISPR-Cas9 is used to understand the function of the gene through deleting or tagging. Deleting or knocking out the gene through CRISPR-Cas9 helps to understand what the absence of the gene does to the organism. Tagging the gene with CRISPR-Cas9 is used to understand the localization of the protein coded by the gene. The purpose of this project is to understand the role of AATF in apoptosis using Crithidia fasciculata. Utilizing CRISPR-Cas9 to modify the gene locus, the gene function will be characterized using tagging and knockout studies. A sgRNA and a drug repair cassette will be generated using fusion PCR that combines a 500 base pair homology arm on either side of the drug-resistant marker. The drug-resistant marker used is puromycin and blasticinin. After the constructs are amplified, they will be transfected into Crithidia fasciculata, followed by a drug selection and screening of successful transgenic parasites. We currently have generated and amplified the drug repair cassettes seen in picture A and the homology arms seen in picture B. These have been generated for tagging studies, and the drug-resistant cassettes for knockout studies have also been created but have not been shown in this poster. Fusion PCR to combine the above fragments shown is still in progress, and future work will focus on transfecting the homologous repair template into the parasites, followed by drug selection and screening.